All right, let's get it started by talking about the homework. So I gave you plenty of time to do this homework because it involves reading of an original paper. So you have time to spend your spring break until next week to submit a short video discussing about this paper, or you want to write a paragraph essay. I want you to explain the idea of algorithm design. Just imagine yourself as the author of that paper. When you start to plan this study, remember we have 3 circles, right? So tell me what the first circle is about. If you were here last week. The biology on the top, because we are biologists. We want to start from this bio problem. And in this reading, and you have to see, okay, what kind of biology question they want to have. I don't know when they come up with algorithm, the idea about this paper. They started probably from a biology question. Then they borrowed algorithm, right? This is A. Algorithm, then you have data in this circle. So this class is teaching about all the algorithm behind those classification, PCA, et cetera, But I want you eventually to have this biology question, then you borrow the algorithm. But the algorithm they use in this paper is not straightforward. It's not a classification. I have a biology question was separate cell from diseased or healthy control, but they used opposite way. So we just try to find, okay, which gene can help me to explain these 2 categories, which cell type. So they use the algorithm, but in a different ways of thinking. So I want you to have those, learn this kind of ways of thinking, and later probably you have some good idea, and you know you can rotate your data points and to achieve something. Although you probably don't know how to do this, but you know the principle. You know, okay, I want to ask my computer science classmates, and please, I want to rotate my cloud of data points in such a way to achieve something like that, because I want to use it to answer this question. I have applications, can you help me? So you start to have your dialogue with cross discipline. So that's interdisciplinary collaborations. So this week, we are going to study, I would say this week is also classification. Last week was a classification about the labeled data points. Can you separate them, train your neural network, okay? So this week is a classification number 2 week. It's so important and in machine learning field. But the one topic we are going to discuss this week is about dealing with nonlinear separations of those. The technique we are going to use is still lineage algebra, okay? Although last week we talked about the neural network. Neural network is like a nuclear weapon. You can tackle any of the problem, and no matter how difficult it is, linear, nonlinear. But sometimes you don't have to bother to borrow that heavy machinery to do some of this lightweighted classifier design, because there are so many lineage algebra, the lightweighted machine you can use to tackle the problem. So kernel is kind of a data transformation technique. Allow you to transform your data, make them separable, although they are nonlinear relationship between data points, but after you apply your kernel transformation, make them separable linearly. So that's the idea of today's. Let's see, where is it? So we will start with a support vector machine. So SVM, this is a linear algebra classification technique, widely used. And then to apply SVM, normally you have to use the kernel or feature mapping functions to transform your data. So we will illustrate how this machine is doing and how you can translate some very complex operations. And it seems impossible to separate those data points, but you, after transformation, you can just apply a lot of very basic linear algebra operations, achieve your goal. So it's lightweighted. If you still cannot do this, you can put it onto the neural network machinery, okay? Then the next half hour, we will talk about a new analysis of cell-cell interactions, okay? Many times, almost feel very bored, I believe, their applications, okay? You have your homework, and you know the dot product of the two vectors is a number. This number tells the angle between these two vectors. If these two vectors have its own magnitude, it's not a unit vector, then you have to scale it up with this number, okay? Because multiplication makes it longer. So I want to play a video to further extend the understanding of this dot product. So this video is from a YouTube tutorial about the wavelet signal processing algorithm. But we just chop in between. There's 3 or 4 minutes of talk between for dot products, okay?
Together. Let's move away from functions for now and consider a pair of two-dimensional vectors. Each vector is essentially a list of two numbers, x and y, specifying coordinates. You may remember from high school that for two vectors, we can compute what's known as their dot product. It's a number which is equal to the product of length of the vectors and the cosine of the angle between them. Intuitively, dot product can be thought of as a measure of similarity between the vectors. If we give our vectors personalities, then dot product relates to their relationship, sort of how close they are. When they point roughly in the same direction, the angle is small and the cosine is close to 1, making the dot product large and positive. This means that the vectors are similar and hence quite close. When they are orthogonal, cosine of 90 degrees is 0, and so the dot product is also 0. This means that the vectors are independent and have nothing in common. They don't even acknowledge each other's existence. When they point in a more or less opposite directions, the angle is greater than 90 degrees. The cosine is negative, hence the dot product is also negative. This means that the vectors are sort of anti-correlated and opposite. Importantly, the dot product, which again is just one number, can be calculated from coordinates. The formula for such a quantity is surprisingly simple. You just negative. Hence the dot product is also negative. This means that the vectors are sort of anti-correlated and opposite. Importantly, the dot product, which again is just one number, can be calculated from coordinates. The formula for such a quantity is surprisingly simple. You just multiply corresponding coordinates of the two vectors and sum everything. So for a pair of two-dimensional vectors, you multiply the x's, multiply the y's, and add the two numbers. For vectors in 3 dimensions, you would need to throw a product of z coordinates into the sum. Increasing the number of dimensions surely makes it difficult to visualize what an angle between the vectors would be. But the intuitive meaning of it as the degree of similarity and the formula would remain the same.
So far, so good, right? That's everything you know already. But next is extension of this idea, okay? So you can do this high-dimensional vectors and look at their similarity between these two vectors.
In the most extreme case, a mathematical function which has an infinite number of points can be considered a vector in an infinite number of dimensions. So if we replace the sum with an integral, which is the limiting case of summation, we can define a dot product between the two functions as the integral of their multiplication. That definition by itself can certainly seem alien and intimidating, but when you realize that...
So here the idea is you treat this function as a vector of data. Okay, you have a point here, maybe 0, and then you go up 1, 2, and down a little bit, and then go to 4. So you can have a long vector, but because there's so many points in there, not the points. So 1 is 1 point. But you can think about this whole function as a one data point, but it just have a lot of numbers in there. Then if you have this, like point F and the G, you can start to do this dot product. It will tell you the theta, the angle between these two. That is a similarity. So the dot product between these two functions become a measurement how similar of these two curve. Okay? So now this curve is not a data point, not one single data point, but it's a time series signal. So you compare one signal with another. Same idea, and just borrow the...
It's essentially exactly the same as the dot product between the vectors. It makes the formula much less daunting and more intuitive. So what we computed back then is exactly the dot product between the signal.
So we stopped here. So this analysis is try to move one signal with another, with one signal as a sliding window to compare with another until you found, okay, this part, they're highly similar. So you know, okay, they're some place that you know the similarity becomes extremely high or low. So this is, again, this is idea you want to know the principle of very basic principle, but expand your thinking to some other application areas. Similar here, okay? As we mentioned last time, PCA, LDA, essentially the operation is the same. The only difference is which rotation matrix you at end will take. PCA will display the largest variability among all the data points. But LDA try to find a rotation, try to find a new angle to separate two cloud of data points. Okay? This is another example. You can think, for example, you know how to do the rotation. You can, in some places, maybe you want to do, I want to minimize the variability of one of the cloud, one cluster, and maximize another. But if you tell the computer scientists, then please give me an algorithm that can achieve something like that, they probably can do it using optimization methods. But you just have to come up with those ideas. Okay? I don't know what the example I just talked about, any applications in there, but for example, you know how to do the rotation. You want to minimize the variability of one cloud, of one cluster of data point, but maximize to display another. So you found a special angle. If you have some application on that, then we can do that. Okay. So here is a small example of using dot product as a classifier to separate green dots and the red dots. And then once you have a new data, and we will, using dot product to compute the cosine, and then to look at the sign of this product and to tell whether this new data point belongs to red or green. So here is the idea. You have some red points, and in this side you have green ones. We need several, so we need C1, which is center of this. By the way, they're all in this coordinate system, so we do the vector operation. So C1 is here, that's in the middle, C plus, and you have a C minus, which in another group center. Then you can, so this C1 is a vector, okay? Everything is vector from 0, okay? So you use C1 to minus C plus, and then you get a vector here. So this vector is called W, okay? You have W. And then you can compute easily the center here, C, is another vector. So that's all we need. Then once you have your input data, say X here, X again is a vector. What you can do is to use this vector, X minus C, okay? X minus C. X minus C in the vector operation will give you this vector here. So the algorithm is very simple. So you just X minus C gives this, and then you have to do a dot product between W and C, okay? W and this. So W is here, right? And this one is here. So you, after this, you do a dot product with W. And the sign of this, so is either this positive or negative, it will tell you this X belongs to this category or that category. The idea, I believe, is if you look at this hypothetical, there's a orthogonal line and the cross C, but orthogonal to W, and then the dot product between this and that tells you the angle of this, basically. Then you use this angle to tell whether this point towards this cluster or that cluster. So theta is somewhere here. Okay, the small example is toy example to tell you, okay, it's dot product between the selected vectors. And you can build that into a classifier. Okay, there are geometric meaning behind those dot products. Okay, support vector machine is kind of those things. You do this dot product, and there are clear geometric meaning behind each operation. But it's just more complex than this, okay? Here, we want to consider, by the way, the idea was in 1963, okay? Wapnick, this professor, I believe, his paper is about support vector networks. Okay, that's the original idea. It's a linear classifier. Okay, if you can read this, he proposed this idea. So the idea is like this. You want to select in this 2D space, for example, you want to have an optimized line to separate two class of data. So SVM try to find how to define this line here. We can separate right circle from green cross in different ways. You can use this line, that line, or this line. They can achieve this. So this is like training data. You train the algorithm. So you gave me a line to separate them. This is 100% accurate. But you want to make this defined line more robust, because in future, if you have a new data point comes in, you want this line still be applicable to separate. So how can you do that? The idea is to find so-called the support vectors. Vectors are data points. To support data points which can define the boundary of this separation and make this boundary as wider as possible. Okay, you need to find those data points, so-called support vectors, which in the boundary of separation plane, okay, you try to make this to select correct support vectors to make this plane as wider as possible. Because you gave the buffer as much as possible for future prediction, okay? So this make this separation line the more reliable or robust compared to if you don't consider you use this as a separator, still work on your training data, but in the future, when you do the prediction, you probably have a very high error rate. Okay? So this is the idea. So this is 2D. If you have a 3D, you will define a plan to separate them. If you high D is a so-called even higher dimension there, hyperplane. Imagine there's some kind of this structure to separate your data. Okay. Any questions? So this algorithm was invented in 1960s. So at that time, the computer is not as powerful as today. And neural network, the idea has been proposed, but because of the lack of the computing power, so we don't have workable neural networks can support those separation. So people try to make this linear operation algorithm as efficient as possible to do the job. Okay, the computation involved in this calculate these lines. The requirement of computation will be much small than to compute the neural networks. Okay. So how can we do that? We need a quick way to compute the distance between data points to any given line. So that's the center algorithm. We need to have a quick way to compute the distance from a data point to the line. Because if you define a line, I will compute all the distance from all the data points to the line, and then to find the support vectors. Okay, so how can we compute a point to a line? I can give the answer first, then we try to derive this formula from scratch. Okay. The answer is to do this dot product, the distance, for example, the distance from a point So from a point V here to a line So here we have two vectors, 2D example. So we have data point V. Okay. Have a coordinate as X or Y. Okay. And W is another 2 points. Would say, okay, A and D. Not 2 points, this is one vector, 2 numbers. So this D, the distance between this point to a line defined by W, can be given by dot product So V, okay. C is constant. It's also used in define the line. So the line will be defined by this and a C, of a constant number. This formula is so simple. For any given line you have in the support vector, that figure, you can very quickly compute any given point in this space to the line, the distance. So you make this computation as simple as possible. So how this formula comes from? So on this side, I will explain this very quickly. Okay. So here we define a line. So this line, as example, we have okay. Normally we will write this line as y. So you have y equals some slope, right? So say alpha x plus a constant. A constant here is a 5. So we know this intercept, this 5 here, okay? So you have to 5 here. So alpha is the slope, right? In this example, the slope is, okay, I just made it up. So we know this. This is a definition of this line here. So if you have x0, and x0, y will be 5. When y is 0, x will be -3, okay? So x will be -3, then y will be 5. So this is a definition of this. So we just rewrite this into that form, okay, 5x minus 3y plus 15. This and this are the same. Okay. Now we have two numbers. One is before x and another before y. So this gave our this w here. Okay, and the c is a constant. C is 15. So for this line, we can use this formula to define it. We can use this formula to define. We can use this. To define a line. All right? So if you have a lot of data in there, I have x red and green. I want to separate. I can try different numbers here. I change this to 6, this to -2. I change. Okay? I want to find a best operation, this curve, definition of this line here. Okay, so when I have, yeah, by the way, if you look at the vector of this, will be a vector here, w will be negative. Okay? So this will be w here. If you want to look at the direction or the relationship between w and this line to define, this something like that. And then you have the position where you cross this. Okay, that's this picture. Everything clear? Now, we have this. Now we have our data. Our data is here, okay, x. We want to compute its distance We want to compute its distance to the line here. The distance is the projection of this onto it. This is d. We want to compute this d. This is also, if you extend this w to this way. So you extend this to make it longer, okay? So this distance between x to this line you define is the same as this. So this is the distance. Is that right? This is the distance we want to compute. Okay. Now this distance, this d, is a big D minus another third. So this D is the length of, how to write? this pink line here. This minus that. I would say this is like delta. This whole thing, the big D. So I will calculate this as a D minus this delta. Is that correct? On the screen is this. This part, the whole D, minus this. Okay? Is the distance. So how can you compute the big D? This is a projection of your x onto W. So big X projection onto W. So you know how to do the projection. So X, that's your data. You want to compute it's the distance 4 times this. This is X W cosine theta. That's so. Yeah, I think I defined a V here, okay? So let's see, this point is a V. So I need, so this D is X norm times cosine theta. So how long this vector, the norm is, and this cosine theta. So because we have this relationship, so this big D becomes X dot divided by norm here. Give this relationship. So this is compute for big D along this flat, the projection. Okay, this is a big D. And then we need to compute small delta here, okay? Small delta here is this V. Because V is on the line, so here we just use so we need to know the norm, okay? The length of this. And then how long this V is, the norm of this is this formula, okay? Dot product between V and W divided by the norm of the, okay? That's still the very, very, very basic formula here. Make sense? So V projection. So how long this, the norm of V is. let's see, right. So V norm is we defined as a delta here. If you use the formula, it is W V divided by norm or W. It is clean this. And because let's see. Okay, because this V is on the line, so W times V dot product is minus C. So you replace this. Okay. So put here. So this is the length of this vector. So the distance is a big D minus this length of the V, okay? Then you replace WV with minus C at the end. You get the distance from this point to this line using this formula. W. Yeah, so W and V are the same direction. So cosine theta should be 0, and the cosine theta should be 1, okay? So that's why you write the length of the V in this way. And then you replace dot product dot W and V using the constant minus C because you define the line here. So there's some replacement, just at the end you get this relationship, okay? We move forward. Okay, so we know how to compute the distance from any given data in the space to a line here, as long as you know the W, using W to define the line, and you know the data point, and then you compute all the distance. Okay? Then you will select from one group, from right group, which point is close to this line. So for example, this one is. You will do the same for the green group. You found, okay, this is a closed one. This is a closed one. So you also found those ones. Then you want to try different separation line and also compute for given separation line from my right group, which is a closed one, from green group, which one is a closed one. We want to make this shortest distance, the closed one, the distance as much as possible, as big as possible. Okay, we maximize the distance from each group the nearest data points to the line, okay? We maximize the distance from the nearest points to the line, okay? So that's involved about a lot of trials, but it's easy to try because you only use two numbers or just vector to define this line. So very easy to define this, okay? Because for any combinations of the two numbers, you get a line here. You can very quickly compute distance from all the data points to it. You can easily find which one is close to the line. And you try different lines and to make this distance from a closed point to the line as bigger as possible. Then that's your line you separate the data. Okay, that involved a lot of minimize and maximize. Okay, you try to fit in your data using, but at the end is you using algorithm to guess what numbers you should have. Okay, you should have 500 minus 6 or minus 502. So whatever combinations give you the best fitting for the results. So you make this distance of a closed point to the line as easy compute as possible, then throw away all this formula to the optimization machine. Then optimization will find the answer for you. There are some other details and about how can you compute the width of this margin or this buffer zone. Okay. Turnout is very simple. Okay, at the end. Then they also introduce the idea of the soft margin, and because their separation is not as clear as possible, sometimes you can allow some errors. Okay, you allow this misclassification of this outer layer, but still keep the wider margin of the separation. So there are some of these trade-offs, whether you allow how much misclassification to make this the boundary as wide as possible, or you want to have very stringent accuracy, but you sacrifice the width of the boundary. So there's some balance you can achieve using some soft margin ideas. Okay, you give a number of mistakes built into your likelihood function. So you want to minimize this, but you have another term that how many mistakes allow you to take. Okay, so this. Different. Okay, so so hard margin and soft margin is a way to deal with this misclassification idea. And the next idea is if you have something like this, the one cluster is embedded in the center of another cluster. So the soft margin strategy won't work because in this case, you need to have some nonlinear way to separate them. In neural networks, you just add more neurons to make this increased layers of the neural networks. So this linear function by combinations and give you the way to separate them gradually, okay? But the kernel method is to do the transformation to allow you transform the shape of this into something like that. And then you can easily to design a separation margin. All right? So this transformation sometimes not do the dimension reduction, so you actually will have to increase the dimension of your data. So we are going to quickly, using small example to illustrate these points, then we jump into some of these applications. Okay, all this just try to convince you the distance from one data point to the line is very simple, very easy to compute. It's a dot product. Okay, so that's why if you can improve the dot product computation by 1%, that will be huge impact on the whole industry, because neural networks is based on that, everything else based on the dot products. Okay. So if you have so see this 1, this 2. So you have this 2 red points, and then I don't know. So you have another cluster of data as a green cross. So this is like 3, 0, and this is 0, 3, and this is 2, 1, 1, 2. So if you want to do a classifier, so use one single line, there's no way you can separate them easily. You have to use a curve. So, okay, right? So in this case, we want to transform, use a kernel function. Use a kernel function. As a x1 x times y. Want some kernel function like this to transform your data. So when you transform this data, you make this into 0, 3, 0. Make this into 3, 0, 0. Okay. And then you transform this into 1, 2, 2, and transform this to 2, 1, 2. So this kernel function actually, after transformation, you increase the dimension from 2 to 3. And this 3D, and you can plot this 4 data points onto the 3D space, then they become separable. Let's see. X, Y. Now you have Z. The first one is 0, 3, 0, 0, 0, 3, 0. Still here. But these 2 data points become jump out. Okay, jump out 1, 2, 2, and then 2, 2, 1. Okay, you can imagine, and this these two green dots are still in the 2D space in XY because their Z is 0, but these two right has jumped up into Z. Now you can separate the data, use a plan here and to cut through. Same idea illustrated here. You 3D after the kernel function and it become 4D. 2D becomes 3D. This is another example, and also not separable before transformation, but after. Same idea. So you have two red points here, okay, in the corner, and two green here. The transformation is also product xy, so xy are the coordinates in okay. Then you have z, make this separable. The other types of this kernel function and apply in different scenarios. Again, the whole idea about kernel method application in SVM is to do the transformation, to do this kernel function transformation before you apply SVM. SVM is linear. Found the straight line, separated data, found the hyperplane to separated points. But if you don't do this kernel transformation, you probably don't have separable data points. But after that, you do this transformation. So this another example, it becomes separable. All right? Gaussian kernel we used quite a lot. In this case, we found the center of the data and then compute the distance from all the data points to the center. So the example is like this. We have some data in the center, okay? Then you have a lot of data outside. We need to find the center here, okay? Then you compute the distance from, let's use this orange. You compute the distance to the center, a lot of small distance. Then you project your distance into this Bell normal distribution curve. If you have distance, small distance, okay, then you project it down to higher peak. If you have long distance, large, you project to the tail of this curve, okay? That make them separable after transformation. You're adding z, depends on how you're close to the center. The close data points will be adding z with higher peak, while the data points which far away from the center will just add a small number based on this curve. And then you separate your data into this shape. Okay, so there's small videos here. Let me play. Okay, so this process is operate as I described. For the larger one, you make the same idea, right? Okay. There's a name for this. This is called the kernel feature space. You have original space of your data, okay, input space. But after you put all the points, go through the kernel function, and you project your data into this hyper kernel feature space. That's a language you want to discuss with the people with machine learning background, okay? You can see, okay, we separate the data after project them through kernel function to the feature space. That's the language they use. Okay, that's what that means. And there are some people call kernel trick because after some of these selected kernel functions, and you can compute the distance, the pairwise dot product between 2 points before and after projection are the same. So it's not very important for us, but it's just a way to do this computation quickly. In the separation, no matter in the input space or feature space, a lot of those operations involved in the dot product between data points. You want to compute the distance, for example, between data. But if you before and after the operation, the dot product are the same, so you don't have to do the feature transformation explicitly. You just compute the dot product between data, and they are the same before and after transformation. So you don't even have to transform your data, because eventually you don't have to. You still use dot product after transformation between data points to compute to do this computation. That you don't have to if you have this trick. Okay, so some of the examples to show you the idea, but the whole lecture can be expanded to the whole semester lecture in computer science. I do have a link in there, discuss about all the kernel functions, using applications, scenarios, different. But we just introduce the idea and terminologies, okay? And also we can do kernel PCA, all right? If you do original PCA, to do the dimension reduction for this, and you will basically crash all the data points into the 1D, and there's no separation between them. But if you do the kernel the transformation first, okay, the kernel transform this, and give the PCA, they will be separatable. Still do the same, but it's on the feature space. So this is the example we will play with. If you do PCA on this, you get something like that. Okay, if you before, and if you apply the kernel, this is like a kernel function. We use the Gaussian kernel I just described, and then you run PCA for the kernelized data, and then you separate them. Yeah, yeah, to help you to separate the data. There are nonlinear relationship between them. Okay, 5 minutes break, and then we finish off the classification. Okay, we move to another topic. Okay, so in single cell study, we always talk about the heterogeneity. Okay, people say, okay, we use single cell data to understand the heterogeneity of the single cell population, and probably in that way, biologists are using quantitatively. But how can we quantify this heterogeneity in a way that we can compute something like that? So if I gave you a population, okay, it's a group of animal or whatever individuals, or in our study will be the cells, okay? So one group is something like this. Another group will be okay. So in this case, it's 100% red. In this case, 50-50. Okay, which group have a higher level of the heterogeneity or heterogeneity? That group, certainly. If we're talking about two different clusters, this will be the maximized heterogeneity you can get. Because heterogeneity was defined as if you randomly pick two individuals from a population, the likelihood they are to be different categories. So in this case, whatever random number, two of them you pick, they're all the same class. So the heterogeneity in this level is a 0, okay? 0. And in this case, we will define as 5 point times. You have heterogeneity level 0.5. And I said that's in between. You probably can have, see, 70% of this, 30% of that. So that will be 0.7 times. Is that right? So if you have 70%, %30 of that, then this is in the middle. So that's the highest you can achieve heterogeneity. This is lowest. So we can use this product to define the level of heterogeneity. So which two product will be the fraction, the p. This will be 1 minus p. So our formula for heterogeneity is p times q, basically, okay? So p is a fraction, and the q is 1 minus p. So that's a formula you want to remember. And normally people will put a 2 here. Okay? I show you why I put two there, because if you think about this as a box here, and then you have so P and the Q, 50%, 50%. So 2PQ means, so this is P, Q, P and Q. By the way, P plus Q is 1. So this is a ratio, okay? So that's the area of 2PQ. In this example, you have 70%. So that's your P, that's your Q. P, Q. Okay? So 2PQ is the area of this purple region. Okay? So the larger the area, the more heterogeneous you have. So this is just two P and Q. If you have a 3 category, you have a frequency P1, P2, P3. You can still use this formula to do that, okay? So why we need to compute this when we discuss about interaction between cells? Because if you have a cell population contains one cell type and another, something like this, then you have 50 this, 50 that. There's higher chance you have a highest chance you have a heterogeneity among your population that they have interaction. If you have something like this, we are not talking about cell-cell interaction. Or for at least we're not talking about this cross cell type interaction there. Within the cell, and probably you can still define subclusters. Any questions? No? Fine. Heterozygosity. That's what I'm writing on the same idea here, okay? So if you have 90% of this, 1% of that, it's small heterozygosity. So the formula comes from PQ squared, P plus Q 1, and then you factorize this, and then to get this, the same as 1 minus P squared Q squared. But that's the formula. Okay. This formula is for if you have 2 classes. But this formula is more generalized for if you have multiple. You have 3 class, K equals 3, 4 class, K is 4. And the F is what a fraction of each category of the individuals in your whole population. Then your heterozygosity level is this. Okay, for the single cell study, we need to take advantage of the data provided to us. In bulk RNA-seq time, we don't have this separation of a different cell population, different cell types. There's no way we can do cell-cell communication analysis in the data if you only have one sample that's a mixture of all the cell types. But now we can have this analysis, which is one of the main areas which you can take advantage of this technology advance. We talked about this before, okay? We know how to do classification and the pseudo-time analysis or differential analysis, we will get into there. And we will talk about the gene program, okay? Involved in multiple genes, okay? How can you detect the coordinate change of a set of genes rather than just 1-2 gene difference? And in the spatial transcriptome error, we can have information of which cell type are located in the structured tissue. These two are related. If you have information about the distribution, spatial distribution of cell types, you can borrow that information. See, cell type 1, the 2 are next to each other, so the cell-cell interaction we detect is associated with the physical contact, maybe cell type, okay? But now we are going to discuss if you don't have spatial information, how just analytically what you can do with cell-cell interaction detection. It's very important, of course, okay? In the immune system, you have physical contact with your T cell with a cancer cell, macrophages, okay? It's important in the tissue homeostasis, in the development. You need to communicate signal between cells. So if you have your secreted peptides or other factors, and the impact is on the same cell type or same cell, this is called autocrine, and it's on itself. And if you have a paracrine that's from one cell and to give the ligand product to another cell with the receptor, so that's a major relationship that we want to detect those interactions. You can have endocrine, you have those secret factors and through this blood vessel and to different locations in your body, okay? There are many different analysis methods have been developed. They all based on basically similar principle. Number one, they need a database. They need a database between ligand and the receptor pairs. Then they will detect the upregulated expression of ligand in one cell type and they try to find corresponding receptor upregulation in another cell type to make the pair. All these methods are almost the same, okay? Depends on which database they use. They may be slightly different. They're based on the same idea of the product of the P and the Q. So in this case, P and Q is not the fraction. P is the expression level of ligand. And the Q is expression level receptor. So each time you look at a pair of ligand and receptor, you search the database, you know, okay, this ligand and this receptor have some pre-knowledge about their relationship. People knows and from other experiments. And then you calculate the expression level of ligand in one population, cell type 1. And then you know cell type 2 expression of the receptor. So this involves pairs of ligand receptor pairs and also their relationship, not a relationship, their relative expression level in cell type 1 and cell type 2. Okay? Pair and the 2 cell types. That's what we need. Yes?
So in cell type 1, the ligand expression is going to be increased, and in cell type 2, the receptor expression will be increased.
Yeah.
Does that also imply that both of them are increased in both cell types? The expression levels are upregulated in both cell types, or you just need them to be upregulated
In
one
one in each. So in this case, you only look at ligand in cell type 1. And you only look at the receptor expression in cell type 2. You don't care ligand in cell type 2. Yes. Or you want to study the relationship in opposite direction. So this analysis is from source to the target, the direction. So you can switch and look at opposite direction.
So like downregulated in each cell type as well?
Yeah, downregulation. I don't know whether the ligand in the receptor cell type has to be downregulated. Maybe not. But this is like a very straightforward. You look at cell type 1's ligand receptor. Cell type, yes.
So if you reverse the interpreter relationship the other way, would the P and the Q still be the same, or would they swap?
They will swap, change. Yeah. So in order to do this, we need to have a cell type annotation, then compute the average expression of all the genes in different cell types. So we need a table. This table will look like the cell. If you work on 2 or say multiple cell types, you have cell type type 1, 2, 3, 4, for example. Then you have to have a ligand 1. Anyway, so this is just genes, right? So you need to build a table, a summary table. So in cell type 1, you probably have 1,000 cells, but this is the average expression. And then you need a database to know, okay, this is a ligand. This is the receptor of the corresponding receptor. Then you make a pair. Then you are ready to do the analysis. You need a summary table for each cell types and their ligand receptors expression. The database will tell you which ligand gene has interaction with which receptor gene. So that's the information. You probably have one ligand and several multiple receptors, and then you have many different pairs to study with. Then you have to start to score this. Yeah. So then you compute a interaction scores. Okay, you have to define which two cell type I want to study and which pairs of a ligand receptor. So you need this information, full piece of information to define one interaction score. You have to compute. Yeah. So because if you have expression matrix after you finish annotation and you can compute a table like this, then you decide I want to study interaction between cell type 1 and 2, and then which pair. So you need to have 4 numbers to study a pair cell type and a pair of receptor, ligand receptor. You have to do this for all the ligand receptor combinations from a database. Each time you have 4 numbers. What you can do with this table, okay, this table, by the way, is from there, okay, from this big table, right? You can start to focus on 2 cell types, a pair of this, and then you compute a score. This is just 3 times 7. Same idea as this, okay, PQ. So what that means is in cell type 1, ligand expression is 3. Well, this ligand's corresponding receptors expression in cell type B is 7. So the dot product between them, if it's big, it indicates that probably cell type A highly expressed its ligand 1, while cell type B highly expressed its receptor gene, which for the corresponding ligand. Okay. So do we have interaction from a B to A? The product between this are 0, so there's no indication cell type B produce this, will cell type A produce its receptor. There's no interaction score in this way. It's only computed that way. So if you look at these combinations and you found this direction, okay, that's also 21, but that's from the source of this cell type B, where receptor, the receiver cell type is A. So that's the opposite direction. So if you have a condition like this, okay, then because cross cell type, you don't have the product, the score is low, so there's no indication of interaction between these 2 cell types regarding to this pair of the ligand receptor. But if you change ligand 2 to receptor 2, the number may change, so you may have detect other ways of interaction.
So is there a way to set up a cutoff value if you
Yep.
upregulate?
Yeah. Yeah, so this is all relative. This is just simple examples, but how can you compute the average expression of this? You use just a pure average, or you want to normalize data, or want to use relative. So there are many ways to treat process the data first to make this more reliable. You cannot just say, okay, in whole population of the cell type 1, there are just 3 cells express that cell really, really high, put average to the top, but 99% of cells not express that gene at all. So we have to have a way to define those, but the principle for all the packages that showed you, they don't care about the variability. Okay, they take average, basically. So I don't trust any of these algorithms. We are not using them. The terrible ones, cell chat is good. Okay, cell chat, by the way, cell chat is fine. Rubbish, I shouldn't say that, but it's giving misleading results. I really worry about all this. So whatever you use, you can always produce something, but if it's misleading, you waste the resource, waste everybody's time. I hope this can be get rid of sooner or later, but they come to the market early and circulate among biologists. They don't understand the principle, they don't care. The other paper uses, we use it, but it's not really, really not good. So I suggest for now, cell chat is okay as a reference. You can give it a try. Don't use them. The rest of them is rubbish. Okay. We develop our own package. So you can use our methods. Okay. So so where you found the example database. So this is one example, ligand receptor database. They just provide the name of ligand, name of the receiver genes. So you can check the database, you can download the data, okay? Human and select this ligand receptor pairs. They will give you those results. So those are the suggested, if you study these pairs in these two. This is a small example, and we can play with this. That's the number, a session number from this data set. This is the mouse data, I believe, totally. So we just go through the toolbox and t-SNE plots, look at cell types. So by the way, this is skin tissue. We found some macrophages, stellate cells, and endothelium cells, most of the fibroblasts. Okay, so now we have 4 different cell types. There are some knowledge about this interaction, possible interaction, so maybe it's from endothelium cell to some immune cells. So you all know this. Now we just select one pair of this ligand receptor pairs as an example. So this example is this gene and that receptor. Which one is the receptor? It looks like this is the growth factor receptor. So, but some of this underlying biology is related. Now we look at the 2 cell types. We focus on endothelium cell and the stellate cells. And then look at this ligand at CAV1 to its receptor, okay? I can compute multiple samples. For example, in this case, I compute the score between this, and the fibroblast. What I found is the score is higher in endothelium cell to cellular cell. How can I compute that score? It's just product, the expression level, okay? I found this direction has higher value. So I will focus on this as a donor source of the ligand of this, and this as a receptor cell type in the stellate cell. On your left is this ligand gene expression level in all the cells, but I highlight endothelium cell. And you can see looks like it's higher, relatively highly expressed in endothelium cell of that gene. And for this receptor gene, you look at stellate cells, okay? So that's an interaction, and we just assume, again, it's just data analysis. There's no biological evidence, but just look at the data. You have highly expressed in this endothelium cell, and corresponding stellate cell have relatively high expression of this corresponding receptor. So that's the signal we try to detect. Yeah.
Sorry, so the last figure, there's a subclass of endothelial cells that have high receptor. So does that say that's the autocrine action?
You mean here? Oh,
Yeah,
oh, this one here.
like the autocrine.
Yeah, we don't know. We don't know. It could be. Yeah. Yeah, if you focus on inside the subcluster, you can do the same. Yeah, you can treat. See, endothelium cell, I have this subcluster as my cell type 1, and yeah. So it depends on your question. So whether you want to go zoom into that detailed subtypes. So similar, just show using this bar to indicate the expression level. So highly expressed here and relatively high in that. As I said, we have our own pipelines or our own tool, and I believe this is the best. Okay. We have a slightly different idea here. You have an expression matrix, okay? We annotate the cells and separate this matrix into cell type A and B. That's our two cell type we focus on. Then we build S matrix, which is the dot product between average expression between genes. So this is gene by gene. We build a gene by gene matrix. That's a crosstalk between this. So the gene I and the gene J, their expression average in two different cell types and dot product put in here. So we build a huge gene by gene matrix, and each element in this matrix is a dot product of this two genes expression in, okay? In this case, we don't care which one is receptor, which one is ligand. We don't need database. So this method, we can detect potential new unknown ligand receptor interactions. For example, if you have a high number here, that means that gene from cell type A as a ligand and this corresponding gene as a receptor in cell B highly corresponding, the dot product is high, so interaction score is high. Although this pair may not in the existing database, we probably later will infer that's the unknown interaction between them. So once we have this, we also build gene regulatory networks. Okay, again, this is gene by gene from cell type A and cell type B. So this relationship tells you the co-expression of not a set of genes, for example, in cell type A, the ligand and with many other ligands maybe co-express, they all highly express. So we have 1, 2, 3 matrices. One is this correspondence matrix, that's interaction score between cell type 1 and B. And then gene regulatory network from cell type A, gene regulatory network from cell type B. So 3 matrices, and then we project using a neural network way all the genes into a low dimensional space. Let's see. These genes, each gene from the data set have 2 copies. One is its corresponding position from cell type A, and then another from cell type B. So every gene has two numbers on this space. Then we look at gene I and J distance. So remember, gene I is from cell type A, gene J is from cell type B. If the space, the distance between them is really close in this projected reduced dimension space, that means they are highly correlated. Either they have high score here, or they support by other members of the network genes, push them together. Again, these 2 genes are the different genes. They're not themselves. So that suggests there's communications between gene I to gene J between these 2 cell types. At the end, we will just give all the gene pairs which close to each other and the top ones, and just report those. So that will produce quite a lot of these ligand receptor pairs. If you don't want to discover new pairs, you can see, okay, I want to search the database, which pairs has been reported previously in the database. We know, okay, this could be a true ligand receptor interactions. Then you can pick those to study the downstream. But this algorithm will suggest a lot of new ones. And it's incorporated information from the score, interaction score, and also the gene regulatory network of each cell type itself. So there are some powers that we can indicate. And also those scores, we not only compute the average, the product of average, we also consider the variability. Okay, there's combinations of this variability and the mean, so plug in this correspondence matrix. So this is some examples in fibroblast to dendritic cells, and then we can detect those interactions between 2 cell types. Not only the interaction, the pairs of the ligand receptors, but also it's within the cell type, the supporting networks. So which other genes have a co-express or regulation relationship to the receptor genes itself, push that relationship. We can go into the cell type to look at the downstream. So after the ligand bind to the receptor, what's the downstream impact? So we can follow that regulatory pathway, basically. So this is an example using 3 cell types. We have a T cells in here, and also we can go into the T cells, see CCR6 within the T cells, what other genes co-expressed or co-regulated with these receptor genes. We don't have a direction, but maybe after we receive information from the inflammatory dendritic cell, the T cells will pass on this information from the CCR6 into internally those transcription factors or other. So that's the power of our method. Not only look at the simple product of the average expression, we consider the variability, we consider where this expression level come from internally, how the cell type specific gene regulatory networks also play a role within the big picture, okay? That's way how we construct the gene regulatory networks and these corresponding networks, corresponding matrix. So this will be used in this neural network learning to do the embedding. And we can also have, if you have 2 samples, see this is interaction between 2 cell types, cell type A, cell type B in the healthy individual sample. Well, this one will be cell type A interaction with cell type B in the disease, the patient sample. We put them together. So this analysis will detect the change of the interaction relationship. So it may have interaction strongly in healthy individual, but the interaction lost in the patient. Okay? So we can detect those by incorporate this into a huge matrix, like 2 samples analysis. Most recent development we are doing is to look at the spatial information about the location of those expressed sample cell type. For example, this is the green dots from a cancer tumor, okay? Tumor cell type. Well, the purple ones, or light purple, are the stromal cells and some of the immune cells in there, but a lot of different cell types which surrounding this solid tissue. We look at those points which in the interface between tumor and that. We don't care within, we only look at those cells in the interaction, the boundary of this, and then detect stronger signal of interaction between ligand receptor within these boundary cells. And then we shuffle we try the boundary cell within inside to build this background. The contrast between these two will give you a strong signal of which ligand receptor interaction has been enhanced within the boundary. Okay, we can shuffle the label of the cells and to detect the interaction between them, between non-boundary cells, but the signal become weaker, and we know, okay, the one we detect between boundary cells are the real ones. And in biology, you don't have a lot of this ground truth, but in this case, we can, through this contrast between cells, interact to each other, physically contact and non-physically interact, to detect those signals, we believe they're the ground truth. Okay. This is called a boundary detection. We need to have some labels and to classify not only tissue and the immune cells, but tissue immune cells with physical contact. Okay. Okay, that's all. Questions? All right.